Hello and welcome to Nikolai's genetics lessons, and today I got a question from one of my subscribers, and here's a question: what size Punnett square do I need to show the cross between these two genotypes? Please help, my test is tomorrow and I am stuck. There has to be a short way to do this. And actually there are two ways how to solve this problem. Both ways going to be correct, but one would be, I would say, stupid way to solve, and another one would be smart way to solve this problem. So let's first talk about stupid way how to solve this problem, but still it's going to be the correct one. So basically, let's start with just one gene with two alleles. For example, gene A, and parent 1 would be heterozygous, parent 2 also going to be heterozygous, and in the progeny we can expect that ratios would be as follows. So capital A here, capital A small a here, capital A small a here, and small a here. So with one gene and two alleles, we got a Punnett square that is 2 by 2. So let's write down this number. This is 2 by 2, and we have 4 cells. If we are going to get a cross as follows, for example, two genes, gene A would be heterozygous, gene B also would be heterozygous, and we have to cross with another same genotype. In this case, we call this dihybrid cross. We are going to get following gametes. By the way, this is going to be gametes. So one parent, male parent, would produce sperm. So this is male side, and another parent would produce female gametes, and this is going to be egg cells. And inside of the Punnett square would be variants of the diploid organism that these parents can produce. So here we also have parent 1 and parent 2, and now we have two genes, gene A and B. And for example, parent 1 can produce following variants of the gametes: capital A, capital B. So let's list it: capital A and capital B. Another variant would be capital A and small b. So capital A and small b. Next variant would be small a and capital B. So small a, capital B. And the last variant would be small a and small b. So small a and small b. If we build a Punnett square, second parent also can produce 4 types of the gametes. The same genotype would produce the same number of the gametes. So we would have genotypes of the gametes: capital A, capital B, capital A, small b, small a, capital B, and small a, small b. So you see, with two genes we are going to get, now we can also make a Punnett square. I'm not going to fill it because it's going to be time consuming. I just want to show you a pattern. So we are going to have a Punnett square that is going to be 4 by 4, and we are going to have 16 cells here. Let me fill just one. For example, let it be this one. So here is going to be genotype capital A, and small b, small b. So we are going to have 16 such genotypes. Some of them would repeat, but our Punnett square is going to be 4 by 4. So let's list this number. so it's going to be with 2 genes. Our Punnett square is going to be 4 by 4 and going to have 16 cells. So 2 genes and 1 gene here. So now we have 4 variants here, 16 variant here, 16 variant here, and you see also a pattern. If we have here 2 by 2, here we have 4 by 4. Now we are not going to write down another variant with 3 by 3. Now let's just do it mathematically. So if we would have 3 genes, our Punnett square is going to be 8 by 8. So we'd have 64 cells. And so these numbers would just grow. We have to multiply by 2. And this is number of genes. And let's return to our problem. How many genes we have here? 1, 2, 3, 4, 5. So let's continue with say 4 genes. We are going to have 16 by 16, and this time this is going to equal to a Punnett square with 256 cells. So once again, this is example with 4 genes and his number of the cells in a Punnett square. And Punnett square is going to be 16 by 16 on each side, 16 variants of the gametes. Now let's continue. We have 5 genes. So that's going to be 5 genes, and our Punnett square is going to be 32 cells by 32 cells, and would give us 1024 cells in such a Punnett square. So you can say that this is going to be an answer with 5 genes. We can build such a Punnett square. But this is going to be not the very best way to solve this problem, and probably this is not the answer that your professor is looking for. Now let me show you some more examples. Imagine that instead of 2 heterozygotes we have a parent 1 that is going to be heterozygous and parent 2 that is going to be homozygous recessive for the gene A. So once again, let's build a Punnett square. So parent 1 heterozygous, parent 2 homozygous recessive. And in this case, let's see what we are going to get. So capital A, small a here, small a here, capital A, small a here, and small a here. So now you see that 50% is going to be heterozygous, another 50% going to be homozygous recessive. So ratio would be 1 to 1, or we also can say 1/2, and 1/2 would be homozygous recessive, or we can say also 50% would be heterozygous and 50% would be homozygous recessive. So all these 3 answers would be correct answers. But is there any other way that we can get the same ratios, but our Punnett square is going to be smaller? Of course. Take a look one more time. Parent 1 is heterozygous, and parent 2 is homozygous recessive. So, as you see, the same example. But let's think parent 1 and parent 2. Parent 1 can produce two types of the gametes. The first one would be dominant allele A, and the second variant would be recessive allele A. But parent 2 only can produce one type of alleles. So we can make a Punnett square that is going to be slightly different. So parent 1 have two variants of the alleles, and parent 1 have only one type of allele, that is recessive allele A. Now let's build a Punnett square, and we would have two variants of the genotypes here. It's going to be capital A, small a, and small a, small a. Now let's think about the ratios. We have, one more time, 50% that is going to be homozygous recessive, and 50% that is going to be heterozygous. This is the same as 1/2 and 1/2. This is the same ratio as 1 to 1, and the same as 50% to 50%. And as you see, this is absolutely the same what we got here. But here is our Punnett square is 2 by 2, we have 4 cells, and here our Punnett square is 2 by 1. But our answer is still the correct answer. So this is going to be the way how we can write down the same Punnett square, but this time this is going to be more compact way how to do it. Once again, so this is going to be, say, sperm, if it is going to be a male parent, and if it is female parent, this is going to be egg cells. If it is going to be male parent and this female parent, then male parent only can produce one type of the gametes, which is sperm, and female parent can produce two types of the gametes. So now, if you understand this pattern, let's recalculate. So basically we can ignore those genes that is homozygous with the homozygous dominant or homozygous recessive. These genes doesn't affect our calculations, and we basically can ignore them. We even can cross them out. For example, we can cross out this genotype, this, and for the second parent we can cross out this gene, and this gene. These genes and these alleles doesn't affect number of combinations that second parent can produce. And here gene that is homozygous dominant, two alleles, also doesn't affect number of gametes that this parent can produce. So here, for example, with parent 2, we have only two heterozygous genes. That means that this parent can produce 1, 2, 3, 4 variants of the gametes. No matter how many homozygous genes we have here, we would have only 4 variants of the gametes here. And here we have 1, 2, 3 heterozygotes. That means that here we have 8 combinations. So our answer would be that our Punnett square is going to be 8 combinations of the gametes parent 1 can produce by 4. This is going to be a number of combinations that of the gametes that parent 2 can produce. So our Punnett square is going to have only 32 cells instead of 1024, which we calculated the first time, which is also correct, but as you see, this time it is much more simple, and we even can fill such Punnett square without any problem. So basically our Punnett square have to be 8 by 4. If you're still confused, here's another example, and this should clarify your confusion. Imagine that we have one genotype that is capital A, small a, capital B, small c, and capital D. How many genotypes of the gametes such parent can produce? And if you take a close look, only two variants. Here's the first variant, capital A and capital B, small c and capital D. So let me list it. So capital A, capital B, small c and capital D. This is the first variant of the gamete. And second variant would be small a, capital B, small c and capital D. So small a, capital B, small c and capital D. And this is going to be second variant of the gamete. No any other variants exist. For example, capital A and this capital B, it is the same like in this example. Capital A, capital B, and say this small c still would be the same genotype. Capital A, capital B, small c, and say this capital D, still the same genotype. So those we have 4 genes here, but actually what is important only how many heterozygote genes we have here. So if gene A is heterozygous, then we would have only 2 combinations here. If say any of these genes also would be as heterozygous, then we would have 2 heterozygous genes, and we would have in this case 4 variants of the gametes. So basically we only interested how many heterozygous genes we have. If one locus is going to be heterozygous, then just 2 variants. If 2 heterozygotes, then 4 variants. If 3 heterozygotes, then we would have 8 variants of the gametes. If all 4 would be heterozygous, then we are going to have 16 variants of the gametes. But because in our example, in our problem, not all the loci are heterozygous, so we would have a least number of variants of the gametes. One heterozygous here, and third. So 3 heterozygotes loci would produce 8 combinations. And one more time, second parent only have 2 heterozygous loci, so would produce only 4 combinations. And we are going to have Punnett square, that is going to be 8 by 4, and would have 32 cells. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.